Dear students, let us discuss about types of immune response too. Introduction. The immune response in our body can be divided into two major types, the humoral immune response and the cell-mediated immune response. Humoral immunity refers to immunity that can be conferred upon a non-immune individual by administration of serum antibodies from an immune individual. Antibody functions as the main effector of the humoral immune response. In contrast, cell-mediated immunity can be transferred only by administration of T cells from an immune individual. Both antigen-specific and non-specific cells can contribute to the cell-mediated immune response. Specific cells include cytotoxic T lymphocytes and T helper or Th cells. Non-specific cells include natural killer cells and non-lymphoid cells types such as macrophages, neutrophils and eosinophils. Humoral immune response. As mentioned earlier, humoral immunity refers to immunity that can be conferred upon a non-immune individual by administration of serum antibodies from an immune individual. Antibody function as effector of the humoral response by binding to antigen and neutralizing it or facilitating its elimination. When an antigen is coated with an antibody, it can be eliminated in several ways. For example, antibody can cross-link several antigens, forming clusters that are more readily ingested by phagocytic cells. Binding of antibody to antigen on a microorganism can also activate the complement system, resulting in lysis of the foreign organism. Antibody can also neutralize toxins or viral particles by coating them, which prevents them from binding to host cells. The primary domain of antibody protection lies outside the cell. Humoral immune response could be in two stages, primary response and secondary response. Primary immune response. The first contact of an exogenous antigen with an individual generates a primary humoral response characterized by the production of antibody-secreting plasma cells and memory B cells. The primary response has a lag of approximately 5 to 7 days before antibody level starts to rise. This lag is the time required for the activation of naive B cells by antigen and T helper cells and for the subsequent proliferation and differentiation of the activated B cells into plasma cells. The lag phase is followed by a logarithmic increase in the serum antibody level, which reaches a peak, plateaus for a variable time, and then declines as the plasma cells begin to die. The duration of the lag phase varies with the nature of the antigen. For soluble protein antigens, the lag phase is a little longer, often lasting about a week. Peak plasma cells levels are attained by 9-10 days, and peak serum titers are present by around 14 days. During a primary humoral response, IgM is secreted initially, often followed by a switch to an increased proportion of IgG. Depending on the persistence of the antigen, a primary response can last for various periods, from only a few days to several weeks. The memory B cells formed during a primary response stop dividing and enters the G0 phase of the cell cycle. These cells have variable lifespans, with some persisting for the life of the individual. The capacity to develop a secondary humoral response depends on the existence of this population of memory B cells as well as memory T cells. Secondary immune response. When an individual encounters an antigen for the second time, a heightened immune response is achieved due to activation of the memory B cells. This is called the secondary immune response. In the secondary response, the lag is much shorter, only 1 to 2 days. Antibody levels are much higher and they are sustained for much longer. The secondary response is also characterized by secretion of antibody with a higher affinity for the antigens. An isotype other than IgM predominates. The secondary response is also characterized by secretion of antibody with a higher affinity for the antigen, and isotopes other than IgM predominates. A major factor in the more rapid onset and greater magnitude of secondary response is the fact that the population of memory B cells specific for a given antigen is considerably larger than the population of corresponding naive B cells. More plasma cells are generated in the secondary response and antibody levels are subsequently 100 to 1000-fold higher. Furthermore, memory B cells are more easily activated than naive B cells. The processes of affinity maturation and class switching are responsible for the higher affinity and different isotopes exhibited in the secondary response. Cell-mediated immune response. The principal role of cell-mediated immunity is to detect and eliminate cells that harbor intracellular pathogens. Cell-mediated immunity also can recognize and eliminate cells such as tumor cells that have undergone genetic modification so that they express antigens not typical of normal cells. Both antigen-specific and non-specific cells can contribute to the cell-mediated immune response. Specific cells include CD8 positive cytotoxic T lymphocytes or TC cells or CTLs and cytokine secreting CD4 positive T helper cells that mediate delayed type hypersensitivity or DTH. Non-specific cells include natural killer cells and non-lymphoid cell types such as macrophages, neutrophils and eosinophils. The activity of both specific and non-specific components usually depends on effective local concentrations of various cytokines. T cells, NK cells and macrophages are the most important source of the cytokines that organize and support cell-mediated immunity. Although humoral and cell-mediated immunity have many distinctive features, they are not completely independent. Cells such as macrophages, NK cells, neutrophils and eosinophils can use antibodies as receptors to recognize and target cells for killing. Also, chemotactic peptides generated by the activation of complements in response to antigen-antibody complex can contribute to assembling the cell types required for a cell-mediated response. Cell-mediated immune response can be divided into two major categories according to the different effector populations that are mobilized. One group comprises effector cells that have direct cytotoxic activity. These effectors eliminate foreign cells and altered cells by mounting a cytotoxic reactions that lyses their targets. The various cytotoxic effector cells can be grouped into two general categories. One comprises antigen-specific cytotoxic T lymphocytes or CTLs and non-specific cells such as natural killer cells and macrophages. The target cells to which these effectors are directed includes allogenic cells, malignant cells, virus-infected cells, and chemically conjugated cells. The second group is a subpopulation of effector CD4 positive T helper cells that mediate delayed type hypersensitivity reactions. When some population of activated T helper cells encounter a certain type of antigens, they secrete cytokines that induce a localized inflammatory reactions called delayed type hypersensitivity or DTH. The reaction is characterized by large influx of non-specific inflammatory cells, in particularly macrophages. Effector T cells. The three types of effector T cells, CD4 positive, T helper 1 and T helper 2 cells, and CD8 positive CTLs. Effector cells exhibit several properties that set them apart from knife helper and cytotoxic T cells. In particular, effector cells are characterized by their large stringent activation requirements, increased expression of cell adhesion molecules, and production of both membrane-bound and soluble effector molecules. Each of these membrane-bound and secreted molecules plays an important role in various T cell effector functions. The fast ligands, porphyrins, and granzymes, for example, mediate the target cell destruction by the CTLs, membrane-bound TNF-beta and soluble interferon gamma and GM-CSF promote macrophage activation by the T helper 1 cells, and the membrane-bound CD4 ligands and soluble interleukin-4, interleukin-5 and interleukin-6 all play a role in B cell activation by the T helper 2 cells. Cytotoxic T lymphocytes or CTLs are generated by immune activation of T cytotoxic cells. These effector cells have lytic capability and are crucial in the recognition and elimination of altered cells, for example, virus-infected cells and tumor cells, and in graft rejection reactions. The CTL-mediated immune response can be divided into two phases. The first phase activates and differentiates naive TC cells into functional effector CTLs. In the second phase, effector CTLs recognizes antigen class 1 MHC complexes on the specific target cells, which lead them to destroy the target cells. The granules in the CTLs contain 64 kilodalton monomers of pore-forming proteins called porphyrins and several serine proteases called granzymes or fermentins, which mediate target cell damage. Immediately after formation of CTL target cell conjugates, the Golgi stacks and storage granules reorient within the cytoplasm of the CTL to concentrate near the junction with the target cell. Porphyrin monomers and the granzyme protease are then released from the granules by exocytosis into the junctional space between the two cells. Granzymes enters the target cells in two ways: by porphyrin pore formation and by the porphyrin assisted pathway. Once it enters the cytoplasm of the target cell, granzyme B initiates a cascade of reactions that result in the fragmentation of the target cell DNA into oligomers of 200 base pair. Within 5 minutes of CTL contact, target cells begin to exhibit DNA fragmentation. Natural killer cells. They are large granular lymphocytes. They show non-specific cytotoxicity and makes up 5 to 10% of the recirculating lymphocyte population. They are active against viruses, intracellular pathogens, and tumors. They produce a number of immunologically important cytokines and play important role in immune regulation and influence both innate and adaptive immunity. NK cells are the first line of defense against virus infection, controlling viral replication during the time required for activation proliferation and differentiation of CTL-P cells into functional CTLs at about day 7. Killing by NK cells is similar to CTL-mediated killing. The cytoplasm of NK cells contain numerous granules containing perforins and granzymes. Unlike CTLs, which need to be activated before granule appears, NK cells are constitutively cytotoxic, always having last granules in their cytoplasm. NK cells employ two different categories of receptors, one that delivers inhibitory signals to NK cells and another that delivers activation signals. It is the balance between activating signals and inhibitory signals that is believed to enable NK cells to distinguish healthy cells from infected or cancerous ones. Antibody-dependent cell-mediated cytotoxicity. A number of cells that have cytotoxic potential express membrane receptors for the Fc region of the antibody molecule. When antibody is specifically bound to a target cell, these receptor-bearing cells can bind to the antibody Fc region and thus to the target cells and subsequently cause lysis of the target cells. Although these cytotoxic cells are non-specific for antigen, the specificity of the antibody directs them to the specific target cells. This type of cytotoxicity is referred to as antibody-dependent cell-mediated cytotoxicity or ADCC. Among the cells that can mediate ADCC are NK cells, macrophages, monocytes, neutrophils, and eosinophils. When macrophages, neutrophils, or eosinophils binds to a target cell by way of the Fc receptor, they become more active metabolically. As a result, the lytic enzyme in their cytoplasmic lysosomes or granules increases. Release of this lytic enzyme at the site of the Fc-mediated contact may result in the damage to the target cell. Conclusion. In conclusion, I would like to state that both the humoral and cell-mediated immune response play a significant role in eliminating any pathogen in our system. In the humoral response, the antibodies play a significant role to eliminate the pathogens present in the body fluid by various mechanisms. But once the pathogen enters our body, the cell-mediated immune response works to eliminate the infected cells. It also helps in eliminating the transformed cells like cancer cells. Thus, both the immune response work hand in hand to eliminate any foreign molecules from our system.